Okay, so picture this. You drop a living organism into a contaminated river, and 6 months later you fish it out, sequence its DNA, and it just tells you exactly what toxins were there, in what order, and when. Like a receipt, printed in actual DNA.
That sounds completely made up.
That's the thing. It's not. Like, this is happening in actual labs right now, and almost nobody outside synthetic biology knows about it.
It's called retron-based living biosensors, and I promise by the end of this, you're going to be equally amazed and slightly unsettled. Welcome to KPsphere Signals. I'm Brian.
I'm Rachel, and Brian sent me this research at 11 p.m. last night with just a skull emoji, so...
I'm John, and honestly, the skull was accurate.
It was very accurate. Let's get into it.
Okay, so before we get to the wild stuff, what even is a retron? Because I had never heard this word before 2 days ago.
Okay, so the wild thing about retrons is they've been sitting in bacteria since the 1980s, and scientists basically just ignored them for 30 years.
30 years. They were discovered in the 80s, classified as these weird little bacterial genetic elements, and then just kind of shelved.
What changed?
Okay, so what they actually figured out pretty recently is that retrons have this reverse transcriptase enzyme inside them, which means they can take RNA and write it backwards into DNA inside a living cell continuously.
And that's the key word, continuously. It's just producing this little strand of DNA over and over, like a factory that never clocks out.
So the cell is basically writing notes to itself?
In real time. And those notes can be engineered to say whatever you want them to say, at least in controlled lab conditions, which is where most of this work is still happening.
Which is where this goes from cool biology fact to, wait, this is a technology.
Okay, walk me through how you actually turn that into a sensor. Like mechanically, what's happening?
So there are 3 layers. Layer 1, you wire the retron to a promoter. A promoter is basically an on switch for a gene. You make that switch respond to whatever you want to detect. Heavy metals, a pathogen, radiation, whatever.
So the environment pulls the trigger and the retron fires.
Exactly. Layer 2, on when it fires, it generates this little custom DNA barcode, unique to that event, that sensor, that moment.
And layer 3 is where CRISPR comes in, right?
Layer 3, CRISPR-Cas integration, specifically Cas1, Cas2, physically inserts that barcode into the cell's own genome, permanently.
So the cell is literally editing its own DNA to remember what happened to it.
That's genuinely insane.
And then later, could be weeks, could be months, you sequence those cells and you can read the whole log. What happened, in what order it happened.
The order part is what breaks my brain, honestly. Most sensors just tell you this thing was present. These tell you whether the mercury showed up before or after the bacterial bloom. That's a completely different class of information, at least in principle, assuming the sequencing and decoding holds up.
Okay, so who actually cracked this? Because someone had to be first.
So the origin story here is great. There's this lab, the Shipman lab at Gladstone Institutes, and they published a paper in 2021. Buttery, Klein, Lear, and Fishman are the authors. And it demonstrated for the first time that you can record the order of gene expression events in DNA.
And this is now cited as the canonical temporal recording paper. Like when people in this field talk about the moment it became real, they cite that paper.
That's a big deal. That's like the Cas9 moment for this whole subfield.
The report literally calls it that, the field's Cas9 moment. And then Schubert, Goodman, and Wanier, same year, 2021, published in PNAS showing you could use retrons for high-throughput functional screening.
2 foundational papers in the same year. That's how you know a field just hit an inflection point.
Okay, so we've got the Shipman lab at Gladstone, Church's lab at Harvard, Wanner at Yale. These are the US players. What's the global picture look like?
So we looked at 165 sources, 74 papers, 51 funding records, and the country breakdown is interesting. US leads with 55 sources, UK is second with 20, then Japan with 9, South Korea with 8.
But here's where it gets interesting. The US dominates the foundational science, but South Korea and China appear to be scaling faster on the applied engineering side.
Define applied engineering in this context.
Okay, so Lee and Kim at POSTECH, that's a top Korean research university. In 2023, they demonstrated engineered retrons that produce ssDNA specifically designed to bind proteins, not just record, but interact.
And the report flags that one as having direct dual-use implications, which is the polite research way of saying, yeah, defense agencies are paying attention.
Okay, I want to come back to the dual use thing because that's a whole conversation. But first, where's the money coming from?
So we tracked $9.3 million in directly attributable funding, which sounds like a lot until you realize this field might be the next CRISPR.
9.3 million is basically a rounding error compared to where CRISPR ended up.
That's kind of the point, though. The report describes this as pre-Series A, pre-IPO territory. The foundational IP is still being filed right now.
Mississippi State got $797,000, Northwestern got $678,000, Clemson $551,000. These are government grants, mostly, flowing into university labs.
So this isn't big pharma money yet. This is still government-funded basic research.
Which is what CRISPR looked like in the early foundational years, before the licensing war started.
The report gives this field a technology readiness level of 3 to 4, which on a scale of 1 to 9 means we've proven it works in a lab. We haven't proven it works in the real world yet.
Okay, so what does real world even look like for this? Like, where would you actually deploy a living biosensor?
Okay, this is where I kept stopping while reading this. The potential applications are so varied, it's almost hard to believe they're all pointing at the same technology.
Give us the list.
Environmental monitoring. The vision is, you drop engineered bacteria into a watershed, come back in 3 months, sequence them, get a complete pollution log. Not just was mercury present, but mercury spiked on this date, then dropped, then this other contaminant appeared. That's what the technology is being designed toward.
That's genuinely useful if it holds up outside the lab. Like, that's the promise of replacing a network of physical sensors with microbes.
Self-replicating, self-powered, potentially zero-maintenance microbes, assuming the chassis robustness problem gets solved.
Then there's in-host diagnostics. The concept is your gut bacteria keeping a log of inflammatory events, antibiotic exposure, pathogen encounters. You sequence a stool sample and get a timeline of what happened in your GI tract over the last month.
Okay, that one's weird, but I can see where it's going.
And then there's the one that made me stop reading.
Which is?
Denied area biosurveillance. The report literally says, and I'm quoting here, persistent biosurveillance in denied environments, meaning you seed a location with these organisms, they record what biological or chemical agents were present, and when you eventually get access, you read the log.
The organisms are the spy, and they don't look like anything. They're just bacteria.
Okay, that's kind of terrifying, actually.
I said that exact thing, out loud, alone in my apartment.
I mean, from a defense perspective, the appeal is obvious. You'd have a passive, self-replicating sensor with no power source, no signal to intercept, no hardware to detect. Whether it's operationally achievable is a whole other question.
Right, but from a who controls this perspective, like the dual-use angle is real.
Very real. And this is actually where the South Korea and China acceleration matters. Because right now the US has the foundational IP, but the applied engineering, making these things actually work in field conditions, that's where POSTECH and others are moving fast.
Okay, let's talk about what doesn't work yet, because I feel like we've been very bullish and I want to pump the brakes slightly.
No, that's fair. The challenges are real.
So the dynamic range problem. The report mentions the msDNA production currently only spans about 2 orders of magnitude. For people who don't know why that matters, that means you can tell the difference between a little and a lot, but not between a lot and an enormous amount.
Which is fine for, was this pathogen present, but problematic for how much arsenic was in this water.
Quantitative measurement is hard. Detection is closer to solved. in lab conditions. Precise measurement is not.
And then there's the chassis problem. Most of this work is in E. coli K12, which is a lab strain. It's not built to survive a river, a soil sample, a human gut.
Right, it's like building a race car that only runs on a smooth track and then wondering why it struggles off-road.
The chassis robustness issue is probably the number one thing standing between TRL4 and TRL5. You need organisms that can handle real-world conditions, temperature swings, competing microbes, nutrient scarcity.
And TRL5 is what the most aggressive labs are targeting by 2028.
2 years, field-deployable prototypes in 2 years, if the chassis and dynamic range problems get addressed. That's not a lot of runway.
But honestly, the pace of progress in this field since 2021 has been faster than most people expected. 2 foundational papers in 2021, applied engineering demonstrations by 2023. That's a fast maturation curve for a field this young.
Let's talk about the regulatory side for a second, because you cannot just release engineered bacteria into a watershed without someone having thoughts about that.
Hard agree. This is an area where the science is ahead of the governance.
Way ahead. And this is actually a pattern we see in every wave of synthetic biology. The tools develop in a relative regulatory vacuum, and then at some point something triggers a policy response.
The GMO crop situation, the gene drive situation.
The original recombinant DNA debates in the 1970s. It's the same arc every time.
The containment strategies matter a lot here, though. You can engineer these organisms with kill switches, auxotrophies, basically making them dependent on synthetic nutrients that don't exist in nature so they can't survive outside controlled conditions.
Can. But will every lab implement those correctly?
That's the question. And the report notes there's currently no standardized biocontainment architecture for environmental release. That gap is real.
And it's a question that gets more urgent as the barrier to entry drops. Right now, this requires serious molecular biology expertise. In 5 years, potentially much more accessible.
Okay, let's zoom out to the investment angle because you flagged this earlier. The CRISPR comparison. Unpack that.
So CRISPR had its proof of concept in human cells. It had university lab demonstrations. And then the licensing races started, companies went public, and the whole sector took off. The report draws a parallel to where retrons are right now.
The foundational IP race in CRISPR was already well underway by the time most investors noticed. The window was short.
And the report explicitly says the window here is 18 to 24 months.
Which started now-ish, like the clock is running, according to the report's framing.
The difference is scale. CRISPR had therapeutic applications that were worth hundreds of billions. Retron biosensors, their near-term killer app, might be in environmental monitoring and defense surveillance, which are smaller markets but also markets with more predictable government spending.
That's actually a more stable base than therapeutics in some ways. Government contracts don't disappear in a market downturn.
And the therapeutic applications aren't off the table. In-host diagnostics is a potentially large market if this works at the required scale.
The pitch in retron biosensors might be persistent environmental intelligence infrastructure, which is a less sexy pitch, but potentially a more durable business. If the field delivers on TRL5.
I want to go back to the geopolitical angle one more time because I think we undersold it.
Please, yes.
The US has 55 of these research sources. The UK has 20. South Korea has 8. China has 7. On the surface, that looks like US dominance.
But the US dominance is in foundational research, papers, theory, first demonstrations. The postdoc work in 2023 on protein-binding ssDNA is more applied. That's closer to a product.
And China's 7 sources are almost certainly an undercount. The report itself flags that a lot of research may not make it into the databases being surveyed.
The report actually notes the unknown category. 36% of sources are from unattributed or unknown origins. That's a big chunk.
So the real picture may be more distributed than the numbers suggest.
And from a national security standpoint, that's something worth watching closely. You have a technology that could passively surveil biological environments and you don't have full visibility into who's developing it.
And you don't actually know who's working on it.
Right.
The analogy I keep coming back to is early signals intelligence. In the 1920s and 30s, radio intercept was this weird niche thing that a handful of military people cared about. By the early 40s, it was shaping the outcome of the war. The window between niche academic thing and geopolitically significant can be short.
Okay, that's a great analogy, and I hate that it makes sense.
Living signals intelligence, bacteria-powered, potentially deployed in rivers and soil and maybe bodies, if the field gets there.
The molecular ticker tape for the physical world.
Okay, so where does this actually go? Like, give me the bold prediction. 2030, 2035. What does this look like?
I'll go first. By 2030, I think it's plausible you have field-deployed biosensor organisms in at least a couple of government environmental monitoring programs. If the chassis and containment problems get solved, water quality, maybe some kind of biodefense application.
I'll take the more aggressive scenario. By the early 30s, someone sequences a biosensor organism recovered from a field environment and publishes a complete environmental log that couldn't have been gathered any other way. That would be the proof of concept moment that brings this into mainstream conversation.
Mine's the uncomfortable one. At some point, we probably have a first regulatory incident. Not a disaster, but something that forces a policy response. An engineered organism that persisted longer than expected, spread further than planned. The field gets forced to reckon with containment standards.
And honestly, that might be the thing that legitimizes the field. Like the recombinant DNA asilomar moment, a voluntary pause, establish norms, come back stronger.
The field that navigates that well becomes the standard. The one that doesn't, that's the story nobody wants to be.
This has been one of the more mind-expanding things I've read in a while, genuinely.
Retrons. They were sitting in bacteria since the 80s, ignored for 30 years. And now they might be the substrate for the next generation of environmental and biological intelligence infrastructure, if the field keeps delivering.
The weird dormant thing in nature that turns out to be the thing. It's kind of a recurring theme in biology, honestly.
CRISPR was also a bacterial immune system that nobody cared about.
Pattern recognized.
If you want to go deep on this, the full research report is up at KPsphere.com. 165 sources, the funding data, the lab-by-lab breakdown, all of it. Worth the read if this topic grabbed you.
We'll be back next week with something that will ruin another perfectly normal topic for you.
That's the promise and the threat. See you then.
Stay curious.